Hi, this is Dr. Cassandra Quave, and in today's lecture we'll be giving you an introduction to the scope and history of microbiology. The slides in this lecture correspond with chapter number 1 of your textbook. So why should we study microbiology? Well, one major reason is that microorganisms are part of the human environment and are therefore very important to human health. As we'll find later in the chapter, they're also important to other things that are of value to humans, such as food and also bioremediation of waste. Microorganisms are essential to the web of life in every environment. Many microbes in the ocean and in bodies of fresh water can capture energy from sunlight and store it in molecules that other organisms use as food. Microbes also serve the important role of decomposing dead organisms, waste material from living organisms, and they can even, in some cases, help to break down some types of industrial waste. They also make nitrogen available to plants. And these are just a few examples of why microorganisms are important and how they interact with other organisms in the environment. Now, the roles of microorganisms are many. First, we know that microorganisms are important to human health, especially when they take on the role of a pathogen. A pathogen is a kind of microorganism that can cause disease in another organism. Microbes are also important to the food chain, and they, again, can help break down waste and also create energy for others. We are filled with microbes all around our body, and especially in our gut. Gut microbes are very important to human health, and they help us to break down our food and to create vitamins that are then absorbed across the intestines. Microbes are also important in the production of different foods. If you enjoy eating breads or yogurt, or beer or wine, or even chocolate, these are all fermented foods that microbes play a key role in creating. Another significant benefit of microorganisms is their ability to synthesize antibiotics. These are substances that are derived from one microorganism that can kill or inhibit the growth of other microbes. Therefore, microbes can be used to both cure diseases as well as cause them. Also, microbes are important as major tools of genetic engineering. In fact, several products important to humans, like interferon and growth hormones, can now be produced in a very economic way by microbes due to the benefits of their role in genetic engineering. Microbes are also being engineered to degrade oil spills and to remove toxic materials from the soil. They can also be used to digest explosives that are too dangerous to handle. So they can serve a major role in helping to clean up toxic waste in the environment. Other microbes can be used to help create energy by turning waste products into energy, whereas others can be used to help create better crops. For example, crop plants can be given certain bacterial genes that produce nitrogen-containing compounds that are necessary for plant growth. And although certain microbes cause disease, learning how to treat diseases, learning how these diseases are transmitted and how to diagnose, treat, and prevent them is of great importance. So microbes are also important not only to understanding how diseases are transmitted, but also in looking for cures. Now, biological research uses microbes as well, and this is due to the rapid growth rate of these organisms. They're easy to grow in the lab and to manipulate for different studies. You can also grow them in very large populations very quickly, and their small size makes them easy to handle. As far as research benefits go, again, you can do research on antibiotics coming from different microorganisms and also help to develop vaccines using these important tools. Another important reason to study microbiology is that it provides insight into life processes in all life forms. Biologists in many different disciplines use ideas from microbiology and use the microorganisms themselves in their research. I'm going to go back one slide just to highlight one more thing. Microbes are very important in research for three reasons in particular. Compared to other organisms, they have very relatively simple structures, and so it's easy to study most life processes in simple unicellular organisms rather than in complex multicellular ones. The large numbers of the microbes can be used in an experiment to obtain statistically significant results at a reliable cost, or at a low cost. So creating 1 billion bacteria costs much less than maintaining 10 rats, for example. And so experiments with large numbers of microbes can give more reliable results than studies with very small numbers of higher organisms. And also, again, because microbes reproduce very quickly, they're really useful for studies involving the transfer of genetic information. Some bacteria can undergo 3 cell divisions in just 1 hour, so the effects of gene transfer can be quickly followed through many generations. Now let's move on to this idea of microbial abundance. We are really the planet of bacteria, and we're just now really beginning to understand how important bacteria are to our planet. Some deep drilling projects have discovered bacteria living at depths that no one had ever believed possible. These are under extreme conditions, both of heat and of different toxic gases. It seems that no matter how far we drill down, scientists have been able to find bacteria living there. And even as we approach the hot interior of the Earth, and where temperature increases with depth, you can find bacteria that live at incredibly hot temperatures. For example, there were bacteria in Alaska that were found living at temperatures of 110 degrees Celsius. Also, at places along the border between the biospheres, materials like oil, hydrogen sulfide, and methane are present. And these bring up with them bacteria from the deep parts inside the planet. Now let's discuss some of the different types of microbes that are found in our environment. The major groups of organisms studied in microbiology include bacteria, algae, fungi, viruses, and protozoa, and all of these are widely distributed in nature. Most microbes consist of a single cell, and again, if you recall, cells are the basic units of structure and function in all living things, and these are discussed in more detail in Chapter 4. Viruses, which are tiny acellular, that means that they lack a cell, entities, are found on the borderline between the living and non-living, and they behave like living organisms when they gain entry into a cell, and these are also studied in microbiology. Microbes can range in size from very tiny viruses that are only 20 nanometers in diameter to very large protozoans of 5 millimeters or more in diameter. In other words, the largest microbes are as much as 250,000 times the size of the smallest ones. Now, as far as bacteria go, among the great variety of microbes that have been identified, bacteria are the ones that have been the most thoroughly studied. The majority of bacteria are single-celled organisms with spherical, rod, or spiral shapes, but a few types can form filaments. Most bacteria are so small that they can only be seen with a light microscope under high resolution or high magnification. Although bacteria are cellular, they do not have a cell nucleus, and they lack the membrane-enclosed intracellular structures found in most other cells. Many bacteria can absorb nutrients from their environment, but some make their own nutrients by photosynthesis or other synthetic processes. Some bacteria are stationary, or they don't move, whereas others can move around. Now, another group of microbes are the archaea, and the archaea are similar to the bacteria, and they belong in the same kingdom called the Monera. There are 3 domains of classification, This includes bacteria, archaea, and eukarya. And these are discussed in Chapter 9. Like bacteria, the archaea are also single-celled and do not have a nucleus. However, they are genetically and metabolically very different from the bacteria. Many of the archaea are known to be extremophiles, meaning that they can live in extreme environments that have very high or very low temperatures, pH, salinities, and hydrostatic and osmotic pressures. As a result of this, their lipids, cell walls, and flagella are very different from those of the bacteria. Most archaea are not proven to cause disease in humans. In fact, many of these are actually important in the ruminant animal digestive tracts. An example of a ruminant animal would be something like a cow. Now, in contrast to the bacteria, several groups of microbes consist of larger, more complex cells that have a cell nucleus. And these that we're going to discuss next include algae, fungi, and protozoa, all of which can easily be seen with a light microscope. Now, many algae are single-celled microscopic organisms, but some marine algae are actually quite large, relatively complex, and known to be multicellular. Unlike the bacteria, algae have a clearly defined cell nucleus and numerous membrane-enclosed intracellular structures. All algae can actually photosynthesize their own food just like plants, and many can move around. And algae can be found in both fresh water and the oceans. Because of their ability to capture energy from the sunlight and the food that they make, algae are also important sources of foods for other organisms. Algae have been of very little medical importance, and only one species, Prototheca, has been found to cause disease in humans. Now, let's discuss the fungi. Like algae, many fungi, such as yeast and some molds, are single-celled microscopic organisms. Some, on the other hand, like mushrooms, are multicellular macroscopic organisms, meaning that they are large enough to be seen without the aid of a microscope. Fungi also have a cell nucleus and intracellular structures. The interesting thing about fungi is that they absorb ready-made nutrients from their environment. And some fungi can make extensive networks of branching filaments, but the organisms themselves are generally non-moving. Fungi are distributed in water and soil, and they serve an important role in the environment as decomposers of dead organisms and waste products. Some fungi can be useful as medicine, either as agents of disease, such as in ringworm and vaginal yeast infections, and others can actually be sources of antibiotics. Now, viruses are acellular entities. Again, this is something that's lacking a cell structure, and they're too small to be seen with a light microscope. They're composed of specific chemical substances, a kind of nucleic acid, and a few proteins. Some viruses can be crystallized and stored in a container on a shelf for years, but they still can retain the ability to invade cells. Viruses are able to replicate themselves and display other properties of living organisms only after they've invaded cells. There are even smaller acellular agents of disease known as viroids, and viroids are different from a virus in that they have a nucleic acid without a protein coating. And prions are a protein without a nucleic acid. Prions that we'll discuss later in class you'll find have been important as a causative agent of diseases like mad cow disease. Now let's discuss some of the eukaryotic parasites. Protozoa are single-celled microscopic organisms with at least one nucleus and numerous intracellular structures. A few species of amoeba are actually large enough to be seen with the naked eye, but we usually study their structure only with the microscope. Many of the protozoa can obtain their food by engulfing or ingesting smaller microorganisms, and most protozoa can move, but a few, especially those that cause human disease, do not. Protozoa can be found in a variety of different water and soil environments, as well as inside animals such as the malaria-carrying mosquitoes. and malaria is a type of protozoa. Now, helminths and arthropods, these are known to be macroscopic, the helminths are, and arthropods are insects and other similar organisms. Helminths have the microscopic stages in their life cycles that can cause disease, and arthropods can transmit these stages as well as other disease-causing microorganisms. Now let's take a moment to discuss taxonomy. It's important that you understand how to properly name microorganisms. The binomial scientific name is composed of a genus and a species name. For example, a bacterial gut species is known as Escherichia coli. Escherichia would be the genus, coli would be the species. It's important to know that there's a specific way that you should write out scientific names. For the genus and the species, both should be italicized or underlined. And the first letter of the name of the genus should always be capitalized, whereas the species should always be in all lowercase. Now, there are different naming systems for viruses, again, because they're not living organisms. And we'll discuss these more in detail when we go into the chapter on viruses. Now, as far as the types of jobs that microbiologists do, they're actually quite numerous. Microbiologists can study many problems that involve microbes. Some microbiologists work on studies about a particular type of organism, for example, the life stages of a particular kind of fungus, whereas others may be interested in a particular kind of function, such as the metabolism of a certain sugar or the action of a specific gene. And there are others still yet that focus directly on practical problems, such as how to purify or synthesize a new antibiotic, or how to make a vaccine against a specific disease. Other microbiologists may be working in the fields of epidemiology, whereas others may look at bioremediation. Bioremediation, again, is a method of cleaning up the environment, controlling insect pests, improving foods, and fighting disease. Bioremediation is a way to clean up the environment, but there are other types of roles that microbiologists may take, such as looking for ways to control insect pests, how to improve foods through food microbiology, or control for foodborne diseases, and so on. Other microbiologists may work in industrial laboratories on the manufacturing of antibiotics, vaccines, or other biological products. Others, such as epidemiologists, may be concerned with controlling the spread of infectious disease and other related public health matters. And this, again, is just a continuation of the last slide. We know that microbiologists play a key role in infection control and helping to track and trace how microorganisms are spreading, and the Centers for Disease Control is one of the main centers of, or the main headquarters for these types of studies. Chemotherapy, again, is the development of different antibiotics from microbes, or antibiotics from other sources to treat other microbial infections. Industrial microbiology has multiple applications, not only in the biosciences, but also in food sciences. And again, biotechnology, such as the creation of new clinical tests for diagnosis of disease, can be important. Now let's discuss a bit of the history of microbiology. We know, in fact, that there has been an understanding of basic microbiology, even dating back to the Mosaic laws found in the Bible that are regarding basic sanitation. Basic sanitation was important in helping to control spread of disease and useful in preventative medicine. Although they didn't quite understand the way that disease was spread, they did implement these types of rules that were helpful in a preventative fashion. Examples of this would be the burial of solid waste material or the isolation of lepers. Another example of knowledge of some of these principles in history would include that of the Greek physician Hippocrates, who lived around 400 BC. He set forth some ethical standards for the practice of medicine that are still in use today. He was able to associate certain signs and symptoms with certain illnesses, and he actually realized that diseases could be transmitted from one person to another by clothing or other objects. The Romans also contributed to microbiology. An example of this is from the 1st century BC of the scholar and writer Varro, and he proposed that tiny invisible animals could enter the body through the mouth and nose to cause disease. The bubonic plague, also known as the Black Death, appeared in the Mediterranean around 542 AD, where it actually killed millions. This was a really key time in Europe, as it actually was a disease that killed both the rich and the poor alike. However, the lack of understanding of disease causation was one reason it took that treatment of the plague was not successful then. Now, until the 17th century, the advance of microbiology was really hampered by the lack of an ability to see these organisms. And around 1665, however, things changed when the English scientist Robert Hooke built a compound microscope, and he used it to observe thin slices of cork. And this is how he actually got the term cell. It's because he compared the little the little units inside the cork, because they reminded him of the cells of monks, these small bare rooms, and that's how he came up with the term of cell. However, it was Anton van Leeuwenhoek, who was a Dutch cloth merchant, and he was a lens grinder, who first made and used lenses to observe the living microorganisms. The lenses that Leeuwenhoek made were of excellent quality and could give magnifications of up to 300 times, and were remarkably free of distortion. He found, when he looked through these lenses, he found what he termed animalcules, and he found them in stagnant water, in sick people, and even from his own mouth. And over the years, he observed all of the major kinds of microbes, including protozoa, algae, yeast, fungi, and bacteria, in different forms, such as spherical, rod, and spiral forms. However, after his death, microbiology did not advance for more than a century. Eventually, microscopes became more widely available and progress resumed. And several people discovered ways to stain microbes with dyes to help make them more visible. Now let's talk a bit about taxonomy and Carolus Linnaeus. Carolus Linnaeus is known for developing a general classification system for all living organisms, whereas the German botanist Matthias Schleiden and the German zoologist Theodor Schwann were the ones to formulate something known as cell theory. And this states that cells are the fundamental units of life that carry out all of the basic functions of living things. And we still apply this theory to all cellular organisms today, but not to viruses. Now let's discuss the germ theory of disease and then spontaneous generation. The germ theory of disease states that microorganisms can invade other organisms and cause disease. And although this is a simple idea to us, perhaps, and is generally well accepted, it was not widely accepted in the past, especially when it was formulated in the mid-19th century. Many people believed that broth left standing turned cloudy because of something about the broth itself. Even after it was shown that microorganisms in the broth caused it to turn cloudy, people still believed that microbes, like worms, such as those of fly larvae or maggots in rotting meat, arose from the non-living things, and this concept was known as spontaneous generation. There was a widespread belief in spontaneous generation, even among scientists, that hampered further development of the science. However, in the late 17th century, the Italian physician Francesco Redi devised a set of experiments to demonstrate that if pieces of meat were covered with gauze so that flies could not reach them, no worms appeared in the meat, no matter how rotten it was. Maggots did, however, hatch from fly eggs laid on top of the gauze. And you can see an illustration of this here on the slide. Though, despite the proof that maggots did not arise spontaneously from the rotting meat, some scientists still believed in spontaneous generation, at least concerning those of microorganisms. Lazzaro Spallanzani was an Italian scientist who was more skeptical, and he actually boiled broth infusions containing organic matter and sealed the flasks to demonstrate that no organisms would develop spontaneously in them. However, this experiment was refuted by critics because they argued that boiling drove off the oxygen, which they thought that all organisms required, and that sealing off the flask prevented its return, and they thought this is why it would not grow in the flasks. So many scientists tried different ways of introducing air to counter this criticism. Schwann heated the air before introducing it to flasks, and other scientists would filter air through chemicals or cotton plugs. All of these methods prevented the growth of microbes in the flasks, but the critics still argued that altering the air prevented the spontaneous generation. However, the proponents of spontaneous generation were finally convinced by the work of the French chemist Louis Pasteur and the English physicist John Tyndall. During the years that Pasteur had worked in the wine industry, he had established that alcohol was produced in wine only if the yeast was present, and he learned a lot about the growth of microbes during this time. Pasteur is famous for his swan-necked experiment, in which he boiled infusions, and you can see a picture here of the swan-necked flask. He boiled infusions in the flasks. He heated the glass necks and drew them out into long curved tubes open at the end. In this way, air could enter the flask without being subjected to any of the treatments that the critics had claimed destroyed its effectiveness. Thus, the airborne microbes could enter the neck of the flask, but they became trapped in the curves of the neck and would never reach the infusions. The infusions from Pasteur's experiments would remain sterile unless the flask was tipped so that the infusion flowed into the neck and then back into the flask. When this happened, the microbes that were trapped in the neck could wash into the infusion where they would grow. Now, John Tyndall also delivered another blow to the idea of spontaneous generation when he arranged sealed flasks of boiled infusion into an airtight box. After allowing time for all of the dust particles to settle in the bottom of the box, he carefully removed the covers from the flasks. These flasks, too, remained sterile. Tyndall had shown that air could be sterilized by settling without any treatment that would prevent the vital force from acting. Now, Pasteur was a giant amongst 19th century scientists working in microbiology, and I want to discuss a few of his other contributions. He discovered that carefully selected yeast could make good wine, but mixtures of other microorganisms competed with the yeast for sugar and made wine taste oily or sour. And to combat this problem, Pasteur developed a technique known as pasteurization, which involves heating the wine to 56 degrees Celsius in the absence of oxygen for 30 minutes, and this was effective in killing the unwanted organisms. Pasteur also made great contributions to the study of silkworm diseases, and during this time he identified 3 different microorganisms which caused a different disease. His association of specific organisms with particular diseases, even though it was in silkworms instead of humans, was an important first step in improving the germ theory of disease. Now, Pasteur also contributed greatly to the development of vaccines. The best known of his vaccine is actually the rabies vaccine, which is made up of dried spinal cord from rabbits that were infected with rabies, which was then tested in animals. And when a 9-year-old boy who had been severely bitten by a rabid dog was brought to him, Pasteur administered the vaccine, but only after a long night of soul-searching, because again, until this point he'd only tested it in other animals. He was not a physician and had never practiced medicine before. However, the boy had been doomed to die, but after delivery of the vaccine, he he survived. And Pasteur later in 1894 became the director of what is known today as the Pasteur Institute that was built in him in Paris. Today, the Pasteur Institute is still a thriving research center for microbiology. Now let's go into Robert Koch's contributions to the as well as Semmelweis and Lister's to germ theory. Now Robert Koch, a little bit of history here, was a contemporary of Pasteur, and he was actually a physician in Germany. He spent most of his time studying bacteria, especially those that cause disease. In fact, Koch was the one that identified the bacterium that causes anthrax. Koch also found a way to grow bacteria in something known as a pure culture. This is a culture that contained only one type of organism. He tried streaking bacterial suspensions onto potato slices and then onto solidified gelatin. However, gelatin melts at room temperature, and some microbes can liquefy it. So Angelina Hess, who was a wife of one of his colleagues, suggested that Koch add agar. Now, if you recall, agar at this time was used in some types of cooking. And this helped to create a firm surface over which the microorganisms could be spread very thinly. And this is how we came to have agar plates. Koch's main achievement, though, was the formulation of something known as Koch's postulates, which provided scientists with a method of establishing the germ theory of disease. His postulates were these. Number 1, the specific causative agent must be found in every case of the disease. Number 2, the disease organism must be isolated in pure culture. Number 3, inoculation of a sample of the culture into a healthy, susceptible animal must produce the same disease. And number 4, the disease organism must be recovered from the inoculated animal. What's implied in his postulates is this one organism, one disease concept, that an infectious disease can be caused by a single organism, and his postulates are directed towards establishing that fact. Another one of Koch's contributions was his development of something known as tuberculin. He was trying to develop a vaccine against tuberculosis. However, he was not successful, but tuberculin is used today in diagnostics for patients to see if people have tuberculosis. Now let's discuss Semmelweis. Philip Semmelweis was an Austrian, and he was and Joseph Lister was English. They were both convinced that microbes caused infections. Semmelweis was famous for recognizing a connection between autopsies and puerperal or childbed fever. What he noted was that many physicians would actually go directly from performing autopsies on dead patients to examining women in labor without even washing their hands. Again, remember, they didn't know that disease could be transmitted by contact with surfaces, even such as the hands. When Semmelweis tried to encourage more sanitary practices, he was actually ridiculed and harassed until he had a nervous breakdown and got sent to an insane asylum. However, Joseph Lister, the his English colleague, who had read of Pasteur's work on pasteurization and of Semmelweis's work on improving sanitation, began to initiate the use of dilute carbolic acid on bandages and instruments to help reduce infection. Now, Lister was also ridiculed, but he had a very resolute will and a tolerance of hostile criticism, and he was able to continue his work. His methods were recognized as the first aseptic techniques, and they were actually proven to be effective because of the decrease in surgical wound infections in his surgical wards. Now let's go on to the field of immunology and some lead contributors such as Edward Jenner, Pasteur, and Metchikoff. Now, disease depends not only on microbes invading the host, but also on the host response to that invasion. Today we know the host response is part of something known as the immune system. A great example of this, one of the early vaccines are that against smallpox. The ancient Chinese, for example, knew that a person that's scarred by smallpox would not get the disease again, and they would take the dried scabs from lesions of people who were recovering from the disease and ground them into a powder that they then sniffed. And as a result of inhaling the weakened organisms, they acquired a mild case of smallpox, but were protected against a subsequent deadly infection. Smallpox was unknown in Europe until the Crusaders returned from the Near East in the 12th century, and by the 17th century it was quite widespread. The wife of a British ambassador to Turkey, known as Lady Mary Ashley Montagu, was the person to introduce a kind of immunization to England. What she did was she would soak a thread in the fluid from a smallpox vesicle or a blister, and then draw it through a small incision in the arm. This technique is called variolation, and was used only at first by a few prominent people, but it later became widespread. In the late 18th century, Edward Jenner realized that milkmaids who got cowpox did not get smallpox, and he inoculated his own son with the fluid from the cowpox blister. He later similarly inoculated an 8-year-old and subsequently inoculated the same child with smallpox. Now today this would be a highly unethical thing to do, but what he did find is that the child remained healthy. And the word vaccina, coming from the Latin word for cow, vacca, gave rise to both the name of the virus that causes cowpox and to the word vaccine. Now, Pasteur also again contributed significantly to the emergence of immunology due to his work on vaccines for rabies and cholera. When Pasteur was studying chicken cholera, his assistant accidentally used an old chicken cholera culture to inoculate some chickens. And what's interesting is that the chickens did not develop disease symptoms. When the assistant later inoculated the same chickens with the fresh chicken cholera culture, they remained healthy. So although this was a case of accidental discovery, Pasteur was very observant and did realize that the chickens had been immunized against chicken cholera. And he was able to reason that the organisms must have lost their ability to produce disease, but retained their ability to produce immunity. And this discovery led Pasteur to look for techniques that would have the same effect on other organisms. And his development of the rabies vaccine was another successful attempt. Now, along with Jenner and Pasteur, there was a Russian zoologist, Elie Metchnikoff, who was also a pioneer in immunology. In the 1880s, many scientists believed that immunity was due to non-cellular substances in the blood, but Metchnikoff discovered that certain cells in the body could actually ingest or eat microbes, and he named them phagocytes, which literally means cell eating. The identification of phagocytes as cells that defend the body against invading microbes was a first step in understanding immunity. Metchnikoff also developed several vaccines. Some of these were successful, but unfortunately some infected the recipients with the microbes against they were supposed to be immunized against. For example, a few of his subjects acquired gonorrhea and syphilis from his vaccines. Now let's discuss virology and chemotherapy. The science of virology emerged after that of bacteriology for the simple reason that viruses could not be recognized until certain techniques for studying and isolating larger particles, such as bacteria, had been developed. Charles Chamberlain, who was a collaborator of Pasteur, was the one to develop a porcelain filter to remove bacteria from water in 1884. He had no idea, however, that any kind of infectious agent could still pass through the filter, but researchers soon realized that some filtrates remained infectious even after the bacteria were filtered out. There was a Dutch microbiologist, Martinus Bedrenik, that determined why such filtrates were infectious, and was the first to actually characterize viruses. Further progress in virology required development of techniques for isolating, propagating, and analyzing viruses. And there was an American scientist known as Wendell Stanley who crystallized tobacco mosaic virus in 1935, showing that an agent with properties of a living organism that also behaved as a chemical substance. The crystals were made up of protein and ribonucleic acid, or RNA. And the nucleic acid of the virus was shown to be important in its ability to infect. Now, viruses were first observed with an electron microscope in 1939. Now, in 1952, there were two American biologists, Alfred Hershey and Martha Chase, that had demonstrated that genetic material of some viruses is actually of another nucleic acid known as deoxyribonucleic acid, or DNA. In 1953, James Watson and Francis Crick determined the structure of DNA, and thus set the stage for rapid advances in understanding how DNA functions as genetic material in both viruses and in cellular organisms. Now let's discuss chemotherapy. Some of you may think of the term chemotherapy and jump to cancer treatments, but in fact chemotherapy is relevant to the treatment of microbial infections, and these are the basis for antibiotic therapy. Now, the Greek physician Dioscorides compiled something known as De Materia Medica in the 1st century AD, and this was a huge work that listed a number of different substances from medicinal plants and medicinal animals used in medicine. This text was later used throughout Europe for centuries as a main source of different medicinal agents. Now, the Swiss physician Aurelius Paracelsus was the one to use metallic chemical elements to treat diseases. He would use antimony for general infections and mercury for syphilis. Paul Ehrlich was the first serious researcher in the field of chemotherapy, though, and he received his doctoral degree in 1878 from Germany. He found that certain dyes could stain microorganisms but not animal cells, suggesting that the dyes or other chemicals might selectively kill the microbial cells, and this led him to the search for the magic bullet, or a chemical that would destroy specific bacteria without destroying the surrounding human tissues. Ehrlich was the one to coin the term chemotherapy. Now, Ehrlich's research initiatives led to the discovery of salvarsan, which was effective against syphilis, and also arsenophenolglycine, effective against sleepy sickness. Now, moving on to the development of antibiotics, in 1917, this came with the discovery, an accidental discovery again, by Fleming. Fleming found that a colony of Penicillium mold that had contaminated one of his cultures of Staphylococcus bacteria had prevented the growth of the bacteria around where the mold was present. So even though Fleming was not the first to observe this phenomenon, he was the first to importantly recognize its potential for treating infections. However, purification of enough quantity of the substance that he then coined as penicillin became very difficult. And the great need for a drug during World War II led to other scientists, such as the biochemist Ernest Chain and Australian pathologist Howard Florey, and other researchers at Oxford University to develop this in larger quantities. Now, while this work was going on, other drugs, known as the sulfa drugs, were being developed. In 1935, protonosyl rubrum, which is a reddish dye that has sulfonamide, was used in treating streptococcal infections. Additional study on this found that sulfonamides were converted in the body to sulfonamides, and much work was then devoted to understanding these drugs. The German chemist Gerard Domack played an important role in this work, and one of the drugs that was named protonosyl saved the life of his daughter. And in 1939, he was awarded the Nobel Prize for his work. Now let's move on to a discussion of future trends. Where are we going with microbiology. So we know that we can use recombinant microbes now to help develop drugs, hormones, and vaccines. And gene therapy has also been incredibly useful. Bacteria have been converted into factories that can produce drugs, hormones, vaccines, and a variety of different important compounds. And also importantly, microbes, viruses in particular, can often be used as the vehicle by which scientists insert new genes into other organisms. And these different techniques are beginning to enable us to produce improved varieties of plants and animals, such as pest-resistant crops, and may even help in the future to help correct genetic defects in human beings. Now, in 1990, there was a 4-year-old girl who became the first gene therapy patient. She had inherited a defective gene that crippled her immune system. And doctors at the National Institutes of Health inserted a normal copy of the gene into some of her white blood cells in the laboratory, and then injected these gene-treated cells back into her body, where it was hoped that they could restore her immune system. The experiment was a success, and the girl enjoys good health today. New information is constantly being discovered, and sometimes supersedes earlier findings. Sometimes new discoveries lead almost immediately to the development of medical applications, such as what happened with penicillin, and which will most certainly occur when a cure or vaccine for AIDS is discovered. Many new bioethical problems can also arise as a result of these advancements. Decisions about AIDS testing and reporting, transplants, cloning, environmental cleanup, and other related issues will not come easily or quickly. However, the future of microbiology does hold exciting developments. One area involves the use of something known as a bacteriophage. These are actually viruses that attack and kill specific kinds of bacteria. Using phages to treat bacterial infections was actually used in the Soviet Union in the 1920s and 1930s. But with the discovery of antibiotics in the and implementation of antibiotics in the 1940s, these fell into disfavor and never really made it into Western medical practice. However, in the Soviet countries, phage therapy has been preferred over the use of antibiotics even today. Soviet troops would carry color-coded packets of various phages with them, specific for the bacterial diseases that they were likely to encounter. In addition to human disease, some agricultural and food problems have also been recently shown to have phage-mediated solutions. For example, Listeria monocytogenes, which is a foodborne pathogen that can live and grow in refrigerator temperatures, can cause bacterial diarrhea that is fatal in up to 20% of its cases. In fact, if you look on the news, you may find examples of Listeria outbreaks on types of cut apples and melons or cantaloupes. Herds of animals can also be protected with phages. The U.S. Department of Agriculture in 2007 approved the use of a sprayer wash that contained phages that target E. coli 0157H7 that should be added to the live animals before being slaughtered. E. coli 0157H7 is a kind of strain of E. coli that causes an often fatal hemorrhagic dysentery, and you can find examples of this also in the news from people that eat contaminated hamburger meat. It's especially dangerous for young children. Other advances in microbiology will likely involve genomics. Genomics refers to microbial genetic techniques that have made possible projects like the Human Genome Project. The purpose of the Human Genome Project was to identify the location and chemical sequence of all the genes in the human genome. That means all of the genetic material that goes into the human species. Work on the Human Genome Project was based on techniques that were first developed for sequencing microbial genomes, which are smaller and easier to work with. And over 100 microbial genomes have been sequenced so far.